Welcome back everyone to a brand new video. After the amazing support I received from the last video, I decided to make another one for you all.
But Alex, what could you possibly teach us today?
But of course, I almost forgot. Today, we will be learning about translation. Translation is the process of converting RNA into protein through the ribosome, an organelle found within the cell right here. Before we jump right into it, let's do a quick recap of how we got here. First, we have DNA in the nucleus of the cell. The DNA will replicate itself through a process called DNA replication. The DNA will then go through transcription. This is when the DNA will convert its genetic code into a copy contained within RNA. Alrighty, that's enough recap. Let's get right into the new lesson. Translation is a process which converts the genetic code contained in the messenger RNA into an amino acid. There are 3 steps to this: initiation, elongation, and termination. Step 1. Initiation. Here we have a ribosome. Within the ribosome, we have 2 sections, the large subunit and the small subunit. The space between the 2 is where the RNA will pass. The ribosome contains 3 sites: the aminosyl site, A site, the peptidyl site, P site, and the exit site. Initiation occurs at the aminosyl site. Here, a codon, which is equal to 3 nucleotides, gets scanned by the A site. The A site must scan the genetic code from 5' to 3'. The start codon is the codon which always starts the genetic code, and it has the code AUG. Once the A site scans the start codon, it knows to begin scanning for more codons. At the A site, we also find transfer RNA, which has an amino acid attached to it, as well as an anticodon. The anticodon is a codon with a complementary code to the one being scanned through the A site. In this example, AUG's complementary code is UAC. By ensuring that both the codon and anticodon match, it ensures that the right amino acid is brought into the ribosome. Step 2. Elongation. As our conveyor belt of mRNA moves on to the next site, the next codon is scanned by the A site. Meanwhile, let's take a look at what happens in the P site. Here in the P site, there is an enzyme called peptidyl transferase. This enzyme cleaves the amino acid from its tRNA and holds on to it. As the mRNA moves along, peptidyl transferase will continue to cleave amino acids from their tRNA. These amino acids join together by using a peptide bond to keep them together. Meanwhile, the tRNA enters the exit site, and the mRNA strand continues to move along. As the amino acid chain grows longer and longer, it starts to take shape of its protein by going through secondary and tertiary structures. Step 3. Termination. While elongation continues to occur, the strand of mRNA which has passed the P site and the tRNA must leave the ribosome. These will pass through the third and final site, the exit site. Eventually though, the chain of codons must end. This is where a stop codon enters the A site. The stop codon forces the mRNA strand to be freed, as well as the chain of amino acids. And there you have it. We have created a protein thanks to an mRNA strand, a tRNA strand, and a ribosome. All research comes from this book, titled Biology 12, as well as my teacher's notes. And as always, thanks for watching.